Hi, my name is Ledesma. I'm an undergraduate student majoring in biochemistry, and today I'm going to present this functional study of the testis-specific nuclear transport rich genes, NTF2R and REN-like in Drosophila melanogaster. NTF2R and REN-like are two RNA-mediated gene duplications that are found in some of the Drosophila species, including Drosophila melanogaster. Their parents are housekeeping nuclear transport genes that are found in the X chromosome of all the Drosophila species, and these retrogenes follow out of the X pattern, which means that they are now located in autosomes. The NTF2R is located in the 2L chromosome, and REN-like is located on the 3L chromosome. Figure 2 is showing some of the functions of the parental genes as housekeeping nuclear transport genes. So, previous studies showed that these retrogenes are testis specific and have been evolving under positive selection. So, we hypothesized that these retrogenes have some spermatogenesis function. To localize these genes, there's the two graphs on top are single cell RNA sequence for both of the retrogenes and the parental genes, which they follow a similar trend. The NTF2 is mostly found in spermatogonia, and the NTF2R is mostly found on the late primary spermatocytes, and this trend is followed by REN-like and REN as well. And the bottom picture is showing some exact localization on early stages of spermatogenesis. We use EGFP protein and DS red fluorescent protein to localize them, and this confirmed that it was found on early stages of spermatogenesis. Previous short-term experiments demonstrated that these genes do not have a spermatogenesis function, so we proposed a long-term experiment, and a previous student started this experiment by using CRISPR-Cas9 technology to produce loss of function mutants, which are KO for both of the retroduplications. We did not see any male sterility, so we started crossing 50 females of the KO for both retrogenes separately with 25 males KO and 25 wild types. And every generation we choose randomly 50 females and 50 males, and we cross them. And every 5 generations we count the number of fluorescent flies, which are the ones that do not have the gene, and this fluorescence is caused by the DS red protein used, and the non-fluorescent flies, which are the ones that are wild type. We did 5 replicates for each of the retrogenes, and figure 5 is showing how the flies look under the microscope. Surprisingly, we found that these genes do have a role in spermatogenesis because we are seeing an increase on the frequency of the wild type allele. This picture showing the 5 replicates behavior. We are currently on generation 31st. They are following the same trend, some changes due to media stress, crossing stress, or media problems that sometimes happen, but they all follow a similar trend by increasing. As well as REN-like, these are the 5 replicates their behavior, and the last figure showing an average allele frequency of the wild type for both, being the green line for DNTF2R and the yellow line for REN-like. We can conclude from this experiment that since the frequency of the wild type allele for both retrogenes have been increasing, which is different from the previous short-term experiment results that another colleague did. We can conclude that these two retrogenes do have a important spermatogenesis function. So this is only strong enough to be seen long term. So now we're planning on study why this is the case, why seen only long term and was not seen short term. I hope you like this experiment and thank you for your attention.